just a science project.
Silent breed is people!
You know, a doctor friend once said the same thing to me. Frankenstein was his name.
It's alive! It's alive! It's alive!
That sounds like something out of science fiction.
Please explain to me the scientific nature of the whammy.
We live in a spaceship, dear.
So? Yes, science!
Program complete. Enter when ready.
Hello and welcome to episode 319 of Science on Top. Today is Sunday, the 2nd of December, 2018. I'm Ed Brown. With me today is Penny Dumsday.
Hello.
Lucas Randall.
Hello.
And clinical research nurse who's just completed a Master's in Bioethics, Joe Benhamu.
Hi, Ed. Hi, everyone.
Master's in Bioethics. That's a useless degree, but anyway.
Is that the arts degree of the biology world?
So. I can't see any situation where you're going to need bioethics. I mean,
no,
I don't know. No. Maybe we'll talk about something later on tonight, but we'll see.
In fact, can we just maybe point out that if people are now sitting at home appalled at our treatment of the guest, we're good friends. This is gentle living.
This is nothing.
And truth is a defence.
I know many politicians who would agree with you.
But before we go on, if you've heard the last few episodes, you'll know that we recently lost a very good friend and a fan of the show. Penelope Green was a keen supporter of critical thinking and evidence-based medicine before she unfortunately succumbed to lung cancer last month. So in her memory, we are donating all the Patreon money we raise for this episode and the next 8 to 2 of her favourite charities, the Fred Hollows Foundation and Doctors Without Borders. So if that's something you want to be a part of, head to scienceontop.com/donate, and we greatly appreciate your generosity. But on the show today, we'll be talking about the big story of the week, the world's first genetically modified human babies. And we'll talk about mitochondrial DNA that can sometimes be inherited from both parents, not just mothers like we previously thought. And of course, that thrilling landing of NASA's InSight robot. Lucas, this was the 56th attempt to send a craft to Mars, and of those, only 26 have been successful.
And most of the rest have been Russian.
No, that includes the 18 or so Russian attempts. But after those heart-wrenching 7 minutes of terror as the craft entered the Martian atmosphere and undertook a series of complex landing procedures, the InSight robot touched down gracefully on a slight 4-degree angle on an area of soft, flat sand called Elysium Planitia. So now what? Why is it there? What are we doing?
Now it's going to do things and stuff.
Cool.
Yep. Basically, this is not a rover. This is a robot that will think of it more as a lab, a remote lab,
stationary lab.
Stationary lab. Its role basically now is to set up shop, deploy its solar panels, which it's done, and they're all working well. It's got bigger solar panels than I think anything else that's been to Mars before, so it's able to generate quite a bit of power, which is really cool. It's got quite a bit of redundant power in terms of what it's collecting, so that's nice. And then what it's going to do is it's going to drill. It's going to send a drill bit down into the ground below where it's stationed, and it's going to use this for a number of different science goals. Predominantly, this is about figuring out what goes on inside Mars. So it's about, for example, what is the core of Mars? What's its size? What's it made of? Is it liquid? Is it solid? We know, for example, Mars has only got a very, very weak magnetosphere. So the magnetosphere on Earth is created by what we believe to be a molten core. And the spinning action of this molten metal core effectively sets up a whole lot of magnetic interactions similar in a way that an electric motor works. So that causes our very significant magnetosphere, and that's important for us. It's important for life because it helps protect us from some of the very damaging types of rays from the sun. It also helps us hang on to our atmosphere. Otherwise, it would be baked off by solar radiation. So we know Mars has a very, very diffuse atmosphere. It's very thin. There is an atmosphere, but it's quite thin. So we want a better idea of what's going on there, because we know that Mars has had, a lot of geological activity in the past. In fact, has the largest volcano that we know of in our solar system, Olympus Mons, which was staggeringly huge and effectively shaped a whole hemisphere of Mars. So there's been activity in the past, but there hasn't been activity that we've seen in the time that we've been watching Mars, and there's not been any signs of any significant activity over that period of time. We don't know whether Mars had plate tectonics, which are obviously very important to Earth. It may have had in the past, but again, there's no evidence of that from what we've seen. But it doesn't mean that it didn't have it. It doesn't mean it still doesn't have it. We just don't know. A lot of what we understand about Earth geology, and Penny's probably better to sort of talk about this than I am, but a lot of what we understand about Earth geology comes from the measuring of certain types of waves as they propagate through the Earth. So when there's earthquakes, for example, they produce all these different types of waves. That's what a seismograph measures. If you've ever seen, movie footage or documentary or something that shows a seismograph, and you see these sort of pens that are drawing on paper as they draw all these different sort of wiggly lines, what they are is a representation of these different types of waves as they propagate through the Earth. And if you have a seismic event on one side of the Earth, you can use the way that waves propagate through the Earth to start to infer information about the makeup of the Earth. You can figure out, for example, what is the density of the region of Earth through which these waves traveled? What is the delay of one type of wave versus a different type of wave, which can tell you things about the makeup of what it traveled through? There's a lot of really interesting stuff that you can get from it. Now, we don't have this sort of information from Mars. I think there's been some seismographs used on Mars before, but nothing like the detail that we should be able to get from InSight with what it's putting down into the ground. So that's one of the key things is how is Mars made up, and what's its current state of its core? How warm is it? Is it possible that there's still geological activity that could be shaping Mars?
So they've got the seismometers there to measure the Mars quakes and the waves throughout the crust, I guess.
Yes.
There are two other instruments on board. Then I think the cool one is the drill, the mole system. Do you want to tell us about that?
So the mole, I had read why it was called the mole, and I can't actually remember why it was. Do you know off the top of your head why it's called the mole?
I assumed it was because it burrows down.
Because it burrows, and I think there was more to it, but I can't quite remember what it was. So it's going to run this probe down into the Earth, and that the probe is more about measuring the heat inside the crust of Mars. Now, it does turn out that where it's landed is quite a sandy region, as you mentioned before in the intro. So the fact that it's sandy might impact on that instrument a little bit, because sand's got air gaps in it, and air is not, well, when I say air, that's probably not the right thing to use in the case of Mars. It has gaps in it, right? So they're not a complete vacuum, but they're pretty damn close to it in the case of Mars. Now, a vacuum is really, really good at not transferring heat. That's why we make thermos flasks with a vacuum tube in them, so that we basically can keep the heat on the inside of the flask, or the outside if you're using it to cool things. So going into sands could present some challenges. If you've ever been at the beach and you've burnt the soles of your feet on the sand, one of the things that you can do is basically plonk yourself down and just bury your feet a little bit into the sand. You don't need to go down very far, but you'll find the sand only an inch or so below the surface is significantly cooler than the sand on top. So with this instrument burying into sand, depending on how deep that sand is, it might actually potentially undermine the goals of this instrument, which is predominantly about measuring heat, and that's to give us some information about how hot the core of the planet is. So that's interesting of its own right. But as you said, it's got a whole. It's actually got several other important instruments on it. So it's got seismometers, as you mentioned before. It also has temperature and wind sensors on it. It has a pressure sensor on it for atmospheric pressure detection. It's got. It actually has a grapple on it, which is, I'll come back to that in a little bit. And it's got obviously various communication stuff going on as well. There's a camera, so it has been sending some imagery back. There's already some imagery that you can look at online from as it started to deploy its instruments and deploy its solar panels and that sort of thing. So there's a bit of stuff going in on there that you can check already online. But yeah, this Mars temperature thing, as I say, this heat probe, the mole, it'd be interesting to see whether the landing situation is going to have an impact on that. And certainly the first thing that occurred to me when I heard that it turns out it's quite a sandy area is how deep is that sand? So yeah, because it doesn't get very deep either. I think. Just scrolling through trying to see what
it goes 5 meters.
5 meters. Okay, thank you. Yeah, I think I saw it in feet, and of course I had a
16 feet, I think.
A brain fluff of what feet were. But yeah, so brain fluff, exactly. So anyway, yeah. So look, what else about it? As I say, that's pretty much its goals is to tell us more about the makeup of Mars. It will be communicating with us via a couple of the orbiters that are already around Mars, much the same way that Curiosity communicated with us, uses the Deep Space Network. So that includes the radio telescopes in Canberra, up in Canberra. Please say the name of the site because I can never say it.
Tidbinbilla?
That's the one. Where we've been, we actually were there for the 7 minutes of terror of the Curiosity landing, so that was kind of cool. But yeah, so it'll be interesting to see what this one comes up with. For me, I mean, Ed and I were talking about this the other day. It's like it's a cool thing for sure, but there's something more emotional, I reckon, about a rover. Something that actually moves. It seems to have more of a personality. You know what I mean? Is that just me or?
No, it's like how when Spirit and Curiosity had Twitter accounts, and you would often sort of identify them as actual robots with personalities and feelings. This is just going to sit there and drill and stuff. It doesn't quite have that same animated feel to it, but I'm sure it will have a Twitter account and it will still be just as interesting. But
right, yeah. NASA and JPL have got better at doing this sort of thing, at raising the public support for programs, and they've really. I think it was really with I think it probably started with Spirit and Opportunity, and then they've done it. They did it for Cassini. They did it for. There's even 2 Voyager Twitter accounts as well that mainly tweet about each other's location in space, which is quite amusing. But yeah, I mean, it's good to sort of start to get people thinking about these instruments. As they kind of we kind of anthropomorphize these things and think of them as having a personality and so forth. Because I know when it was looking like Curiosity might not recover from its recent shutdown, that it was a case of, oh my god, we might have lost Curiosity, which is so sad.
Do you mean Opportunity?
Oppie as well, but Curiosity as well had some issues with some not returning signals and so forth. So yeah, it's always one of those things that we worry about. And the couple of times that we had some of the other probes, do you remember there was just this massive sort of holding your breath period of time of will New Horizons wake up, because it was in that deep sleep for such a long time when it was heading out to Pluto, and it basically it had to wake up on its own at the schedule. It's like, is it going to call home? So But anyway, as I say, we'll see what comes out of this one. We won't get, all sorts of cool imagery like we've had from Curiosity and Spirit and Opportunity before it, and even Pathfinder to some degree, because the thing's not moving. So it can look around where it is, but that's kind of about it. But hey, it's another important step.
I wonder how much of our anthropomorphizing of the rovers has come from what was the name of that movie?
Wall-E.
Yeah. I wonder how much sort of cultural effect that's had in terms of how we view them.
I reckon it would have, and like I don't know if you remember, there was another movie back when we were kids, Joe, which
Short
was
Circuit.
Short Circuit. Not
Short
was
Circuit,
alive.
yes.
Yeah. So yeah.
I didn't actually know if you were going to say that. I just thought this is the best one. It has to be that.
And I mean, it's funny. You have the particularly Curiosity, because it's got that boom on it with the camera that can move around and so forth. It just kind of looks that it seems like a creature, which is kind of cool. But yeah.
Also, it takes selfies. So
it's selfies. Oh yeah, absolutely. Yeah.
And we were talking about Opportunity before and the difficulties that it faced. I haven't heard anything recently. I think the last I heard was middle of November, which was that, no, it hasn't awakened yet after the dust storm that sort of forced it to shut down. There was a false alarm. There was like a blip of data that we thought was from that, but that wasn't actually from Opportunity. So we're still trying to get it back.
Still waiting. And I think the concern was that I think most commentators have sort of said, look, we expect it will come back, right? Because sooner or later, even if it has got a build-up of dust on its panels, which it would have, sooner or later it will blow off, right? Because that's just the cycle that happens. But enough that it would be drawing enough power. But the problem is that they basically said they were going to stop listening for it. And I remember talking about in that episode that I just find that a little bit weird, because we've got, what is it, 3? We've got MRO, and we've got I think there's 3 orbiters that are capable of communicating. It might be only 2, I can't remember. But it's not like they're using dollars listening. You know what I mean? It just seems a little odd to me that they would just go, well, that's it. It didn't get back to us. We're cutting it off.
They gave like a 45-day window where they'll active listening before they would stop. Active listening. They've extended that now through January, but who knows?
Okay. But I don't know, it just still seems a little bit like, quite okay, if you don't get back to us by now, that's it, we're done. We're not friends anymore. I'm unfriending you. This seems a bit severe.
But let's talk biology now and have a look at DNA. We're all made of roughly 50-50 split of DNA from our mother and DNA from our fathers. That's true at least in the nucleus of our cells, our chromosomes. But there's another part of our cells that also has DNA, the cell's power plant, mitochondria. And we've been pretty sure for a long time now that that DNA is always only passed down from the mother. Until, that is, Tao Sheng Huang, a pediatrician and geneticist at Cincinnati Children's Hospital, noticed something unusual in the blood of a 4-year-old boy. Penny, what did he find?
He found, and this is really unusual, that mitochondrial DNA that wasn't just from the mother. So 2 different sets of mitochondrial DNA in this boy, which is just not textbook biology. So what we all learn at school and so on is that, you get your nuclear DNA. So the vast majority of your cells, I should say, has a nucleus which has chromosomes in it, and half of them come from mum and half of them come from dad, and together they not interact, but each one can express perhaps slightly different versions of a gene, and that gives you your unique characteristics. But all your mitochondrial DNA comes from your mother's eggs, and the mitochondria are little structures inside each cell which are really, really important for the way that we essentially, like, long story short, break down our food into energy that we can use. So they're really, really important. They are, you might have heard of them as the powerhouse of the cell in your good old biology textbook. But yeah, they come from the mother. And the explanation for that has been essentially is that if you think about what happens when a sperm meets an egg, the sperm is seen as kind of a little nuclear DNA delivery capsule. Like, it's got its own mitochondrial DNA, but it doesn't really contribute that to the new human. So it's what's in the egg. The egg has its DNA, but all the rest of the egg got inherited as well. So it's got kind of a starter kit of a cell that divides and then becomes the new person. So along with the mitochondrial DNA. And so what's important is that even the authors say, look, this doesn't invalidate the central dogma of how mitochondrial DNA is inherited. It does show, though, because overwhelmingly this is probably not going to affect any of the really fascinating studies that have been done using mitochondrial DNA, or, you know, even our broad understanding of how it works. But it does show that there are exceptional cases where it can happen that a father passes down some mitochondrial DNA to offspring. And I think it was found in 3 families, 17 people all in all. And a lot of testing, because when this has popped up, it's usually been dismissed as a lab error or some kind of error. And so to actually go out there and say, no, we are sure that this is a thing that's happening. Like, it's not just, they found a random report and went on from that. So what does it mean and how could it happen? We're not exactly sure how it happens. It's thought that in the nuclear DNA, so what we usually think of as our genes, there's a little instruction that says to a sperm or to an egg, get rid of that sperm DNA, and a little marker on the sperm DNA that says, this is sperm mitochondrial DNA. This is from the dad. You don't want this stuff. So it can recognize it, and it usually is got rid of because the sperm and the egg do fuse. So, you know, those mitochondria from the sperm that are left will go there. So it's thought that, and we don't know the mechanism yet, but perhaps a malfunction in one of those genes. What does it mean is quite interesting, especially because there are diseases of mitochondrial DNA. So children who do inherit DNA from both have hybrid mitochondrial genomes. They don't just have one kind of mitochondrial DNA, they have more than one. And just from my really basic understanding of genetics, that could possibly be advantageous, especially if there's something wrong with one of the sets of mitochondria. The other one might be able to pick up the slack. That sometimes happens with normal genes. It also seems to be one that can be passed on to offspring. So men who inherited this hybrid genome pass that on to their children, but not always. So I think that's really interesting. And what it might mean is, I guess, as opposed to any potential advantages for the child of having this hybrid DNA, probably, I'm guessing if we know about 17 cases, it's happened in more. Like, most of us haven't had our DNA sequenced. Yeah. It's not some discernible thing that's been obviously popping up. But what the article I read and what I instantly thought of was fertility treatments and this idea of 3-person babies.
Yes.
Yeah. Which is when there's a disease with the mitochondria of the mother, which are the ones that get passed on. And so we've had these situations where you can have an egg from a donor that gets its DNA taken out and the mother's DNA put in and then fertilized with the father's sperm. So essentially there's 3 parents, one donating the egg and the mitochondrial DNA and then 2 donating that nuclear DNA. And if you can somehow get DNA from the father, like, I don't know enough about this to say any informed thing, but it just seems to me that it could open different treatment routes for people who do have these diseases and want to have their own children, which is really, really Interesting. I also do wonder, like, even though they said it doesn't invalidate that central dogma of maternal inheritance, like, that's a really, really overwhelming thing.
Sure.
What kind of consequences it might have had, and how many of these hybrids there are, and if any of them happen to, like, slip into a study and weren't noticed. You know what I mean? Like,
exactly. You'd assume that this is the way it always is. So if anything comes up different, it's either going to skew your results or you're going to ignore it because that's weird. But yeah. It also makes me wonder, why is it that mitochondrial DNA is always, until now, only passed from the mothers? Is that like, I sort of remember, how a baby is a foreign body and the immune system will often see it as different DNA and a possible attack. Is it possibly that it sees the mitochondrial DNA as foreign as well, so the body will scrub the father's mitochondrial DNA? I'm wondering what evolutionary reason there would be to not scramble the mitochondrial DNA as well, that's all.
It's interesting, isn't it? Because, I mean, the impression I've always got, and I'm by no means an expert in this, is it's just the egg is big and has everything. And even though that's not as pronounced with humans, like as with birds, for example, like the egg provides, or the mother, so quote unquote, I don't know why, you know what I mean? Like, because obviously you've got your seahorses and weird stuff going on, but generally the mother is the one that provides the big gamete with all the extra stuff. And I always thought it was, I guess, some kind of, different strategy for mothers and fathers in terms of passing on genes and so on. But yeah, like why wouldn't it combine? One of the articles suggested that it's been thought that like having two sets of mitochondria might make it more difficult somehow for the nuclear DNA to coordinate what was going on if they responded in different ways to commands from the nucleus. I'm just saying that I read that. I don't really understand how that would work, because I've usually thought of, as I said before, in genetics, having slightly different copies of the same thing is usually kind of good. Yeah.
It's complicated, and I only understand a little bit of this, but I've said it before. I love it when we find that a well-established rule is not quite as well established as we thought it was, sort of thing. Our favourite saying is, I think you'll find it's a little bit more complicated than that. And
Did you see me backtracking when I said, and all your cells have... But sometimes I'm like, why can't we just have simple things,
Because evolution is not simple, because biology is not simple.
Why can't I just have my favourite Mother's Day card that I never got, which was a picture of a mitochondria saying, Mum, you gave me something that Dad never could.
And now it's wrong.
Now perhaps 1 in 5,000 dads are, so there you go.
Yeah. Well, let's stick with the biology and the DNA and talk about what I think is the biggest scientific news of this week. Chinese scientist Professor He Jiankui announced he's created the world's first ever gene-edited babies using the CRISPR-Cas9 technique. He claims 2 twins, baby girls named Lulu and Nana, were born to a couple in China earlier in November. He also claims another pregnancy is underway using this method. Professor He claims he replaced a gene in their DNA so that it blocks infection by the HIV virus. There's a natural immunity to HIV in under 1% of the world's population. So he's essentially replaced 2 genes in these embryos with ones from people with this immunity. And Joe, this is really causing a lot of controversy for so many reasons, isn't it?
Oh my! Whoa! I think when this announcement came out, bioethicists around the world all screamed very loudly, as did a lot of researchers.
Yeah.
Because this announcement has tremendous implications, not just for, the people who are sort of, sitting there pondering, the philosophy, the ethical kind of issues around this, but it has huge implications for the scientists who are working on these issues, and who may actually see their work restricted because of what has potentially been done. And I say potentially only because He Jiankui's work hasn't been verified as yet.
And that's the first of the many issues with this, is it wasn't published in a peer-reviewed paper. This was
it wasn't
via YouTube and the Associated Press.
It was announced in a very unconventional way, and so therefore it hasn't been subject to scrutiny, and it hasn't been subject to scrutiny at many levels, not just the scrutiny of the peer review process, but it also hasn't been subject to the scrutiny that any human subjects research should go through prior to that research even being conducted. So there are some fundamental concerns around the ethical validity of this research, and the scientific validity as well. It hasn't been validated whether he has actually achieved what he has claimed to have achieved, and certainly there have been examples in the past of researchers who have made quite extraordinary claims. None of them actually specifically come to mind, but certainly there are examples in history of where scientists have claimed to have done quite extraordinary things, and it has been demonstrated not to be the case. But I have to say I'm a little bit hesitant to think that would be the case. In today's climate, where with social media and the sort of the publicity people get, I suspect that he may have actually done what he claims to have done.
Yeah, I think this is genuine.
Yeah, no, so do I. And I guess in terms of talking about the implications of what's been done here, they're really quite vast, because they go to not just the implications for the research side of it. There's also elements of the rights of the individual, the rights of the babies who have effectively been the subject of this experiment, the rights of the parents in terms of the informed consent they gave, and so on. And I'll go into some of this in a little bit more detail. I don't know, do you have any specific questions, or do you want to just jump into it?
I want to say that it's very easy to label Professor Hui as a maverick and a rogue scientist. And yes, it's been very unconventional and possibly illegal in China, certainly in contravention to an unwritten agreement amongst geneticists that we're not going to start doing this with viable human embryos just yet.
Yeah, that's right. The National Academies in 2015 agreed to a moratorium on germline editing until there'd been further discussion, both within society and within their own communities, and in terms of the development of the technology.
Which is kind of the irony here, is that he made this announcement alongside an actual conference, which was set up, the second ever conference, to discuss that very problem of when is it going to be okay to work on this, what are the procedures and guidelines that we're going to follow with that sort of a thing, when can we start doing it. But it's also not without precedent, this sort of thing. This is not the first time that a radical new technology has been used to create babies that was not announced through peer-reviewed journals or anything, but it was announced through the media and everything. That was, of course, Louise Brown, the first, quote, test tube baby, the first IVF baby. And of course,
what year was Louise Brown born again? I forget what year that was.
1977 or 78.
Yeah,
she was born. Sorry, yeah.
Now, interestingly, so if we look at that in terms of the history of bioethics as a discipline really was in its infancy in the 1970s.
Sorry.
Yeah. So at that time was when the work of some of the sort of really key voices in bioethics were starting to really develop some of the literature around the principles of biomedical ethics. So Beecham and Childress, who are two of the key names in clinical ethics and research ethics and so on. That was when really a lot of that work was starting to come out around the time also of the Beaumont report in America, which all was an offshoot of the Tuskegee experiments. All of these things kind of came out as a result of highly ethically dubious experiments conducted on humans. So while I think absolutely we can look back and say, these things have happened in the past, I think when we look at how we conceived of the ethical issues and how we have come to understand them over time, things have changed hugely in terms of the both the legal and ethical parameters around which researchers have to operate over the last 40 years.
Sure. I guess my point, though, with talking about Louise Brown is that in 2010, the surviving co-developer of that procedure, Robert Edwards, was awarded a Nobel Prize in medicine for that work. And you can't deny that it's been a profoundly successful procedure and has changed the world in many ways. And there's a degree to which sometimes you need those scientists to push the boundaries. But I'm deeply concerned when it comes so prematurely and without any sort of oversight or institutional regulation or anything like that, because this was done on his own time. He took research leave from the institution and paid for it out of his own pocket. And yeah, I'm deeply concerned by this. This is...
I am deeply concerned because there's something, when I look at kind of how this was announced, and also when I look at how he has rationalized both how him and his team chose to target their research in terms of their use of CRISPR. Specifically, so just to kind of give a little bit of background here in terms of the nature of the research. So we've had these two little girls who've been born and have had their genome edited. The purpose of the editing was to protect them from HIV infection, as you said. Mm-hmm. So they reportedly enrolled 7 couples into the trial, and these were all couples where the fathers were HIV positive, and they used the eggs and the sperm from the parents to create embryos using IVF, and they then used CRISPR to deactivate this gene. Now, his rationale for using HIV as the target for this first experiment were what he described as safety and value. And I'm not entirely sure what he means by that, because in terms of safety, I'm not sure whether he was talking about the fact that, this mutation that protects a certain percentage of the population is a known mutation, and therefore if I understand correctly, he was therefore just mimicking that mutation, or whether he was using safety to say, well, he was confident that the technique was safe. In terms of value, if we talk about value, it seems that his rationale for or his justification for this experiment was that it would be of great benefit to society because, so many millions of people could therefore benefit from this. Now, sure, there, you could argue that there are many things that could be done which would be of value to society. Mm-hmm. But the key question when we think about the question, when we think about an ethical question, what we're asking is what ought to be done? What should we do? Okay. So if we're going to say, this particular technique is going to provide value to society, well, society also therefore needs to have a say in deciding whether we think that particular thing is of value. Now, certainly we need the input of experts into that as well. You can't argue that the average person on the street who has no understanding of the technology would agree with that, and that's how we often end up with people pushing back on things like GMO crops, because they don't understand that that the safety has been demonstrated and therefore the value is there. But it's not just up to him. I think it's the hubris that is really, really concerning here, is this idea that he has made this judgment of his own accord. Now, in terms of making this determination, the normal process by which any researcher would be expected to go through is to have their research reviewed by an ethics committee. Now, an ethics committee are a group of people who are tasked with assessing a particular research protocol against a certain set of standards and ethical guidelines that have been put forward within their particular institution or their particular field or country. So the people who make that assessment are not necessarily going to be experts in that field, but they certainly are expected to determine whether the benefits outweigh the risks, and whether conducting that research is ethically justifiable on that basis, and whether the research has scientific merit. Now, he completely bypassed any sort of oversight. And something that I find incredibly troubling from any researcher is a researcher who thinks that they are above having their research evaluated by an ethics committee because they know better. Now, a qualified ethicist, so someone who has a philosophical training and who actually understands the ethical issues, can really engage with a lot of these questions around the implications for society, what can be justified and can't be justified, and so on. So there are just multiple levels by which different people who have expertise or have a stake in giving this kind of, or any kind of human subjects research, the tick of approval, have been completely bypassed. Now, on the one hand, you could say any kind of low-level, low-risk research, you could kind of say, okay, if a researcher, bypassed a very low-level ethical review with something that was kind of an established practice and so on, it's not okay. It breaches certain expectations and standards, but there's no sort of really inherent risk. But we're talking about a first-in-human, fundamentally ethically questionable technique that has been done without any engagement with the sorts of people who need to have oversight of that, including experts within his own field. So it really speaks to me of someone who is really wanting to be the first to do this, rather than someone who is particularly concerned about the impacts for the people who are going to be affected by it.
Yeah. So when he talks about safe and value, I think what he really means is that they're low-hanging fruit, that HIV is something where there's only one or two genes that you need to edit and it's done. It's not something more complicated. Yeah. But the trouble is there are great risks to this. There's on-target and off-target risks. So off-target are the fact that CRISPR is a pretty precise mechanism, but there's the chance that when editing one particular gene, you might also edit a related gene, and we're not quite sure how those genes might be interrelated and how it might affect them.
Exactly, and those sorts of things can potentially result in developmental problems, miscarriage, and I think one of the other risks is of mosaicism, which is that the change can occur in some but not all cells. So one of the articles I read described how an embryo can end up having heart cells that have the change, but liver cells that don't, and we don't really know what the implications of that are.
And it does look like, again, with the limited amount of data that we've got, it looks like one of those babies might have that mosaic issue where not all of the cells have the replaced gene. Then there's the on-target risks, which are, yes, you might reduce the risk of HIV, but those people with the HIV immunity are also more susceptible to West Nile virus and just the ordinary flu. In fact, they're more likely to die from the normal flu. So you're still messing with things that we don't fully understand yet and creating more risks and dangers.
Yeah, absolutely. That's spot on. And one of the things that I found particularly concerning is that Professor Jennifer Doudna, who is one of the pioneers of CRISPR, she was very troubled by the approach that they've taken, because she said that, given the fact that deciding to press on with gene editing in human embryos is such a problematic step to take that if it's going to be done, it really needs to be done in an area where there is a clear unmet medical need and where there is no alternative viable approach.
Yep.
And the point that she made was that if this was done, and I'll quote her here, she said that if this was done to avoid HIV infection, there are alternative ways to prevent infection that are already effective, such as washing the sperm of infected sperm donors to eliminate HIV. And she asks, why would you use this instead of an already established approach? And I find that quite interesting because if you look at clinical research, so where, when you conduct a clinical trial in oncology or in any other area, it is unethical to, unless a person has no other options, it is unethical to offer them an experimental treatment when there is a demonstrated effective treatment. Yeah. And that then goes on to the issues which I'm also very concerned about here, because, we're thinking about this very much in terms of the societal concerns. What is this going to mean for society, the downstream effects, and so on. But what are the implications for these babies in terms of potentially, if they are identified, then they might be treated differently by society. They, if they do develop problems growing up, they haven't had the ability to be able to make a choice about whether they wanted to have been a subject of this experiment in this way.
And the parents have reached a contract and agreement for regular health checkups with Professor Hui. So they're already now locked into giving Ray private information, which he obviously says he's going to respect their privacy in that. They're now long-term experiments. And he says that hopefully when they're 18, they will give consent for continued monitoring. That's a huge thing to expect someone to be a lifelong experiment.
Absolutely. And in fact, one of the big concerns that I've seen others express, and this is one of the first things that I thought about, partly because it's the area that my thesis was on, is around the informed consent aspect of this, which is what did these parents understand about what they were signing up to? So I read that he called his work AIDS vaccine development. Now, well,
it's not a vaccine.
It's not a vaccine, absolutely not. And apparently sort of the process that they went through was suboptimal in terms of how they provided information to these people. And it's difficult enough when you're consenting people to a medical treatment that people are already fairly aware of in society. So, if we talk about chemotherapy or if we talk about abdominal surgery or something that people are familiar with, you can kind of at least to some degree expect that they'll have an understanding, a very sort of basic understanding of what those things involve. These people are being asked to provide consent to a completely experimental technique with potentially limited understanding, not just of what the technique involves, but the implications for their future children. And that is incredibly concerning.
I also wondered about the children's rights to have their own children later on in life. It's kind of like that. You're pretty much guaranteeing that this genetic modification is going to, continue on through generations.
Well, they don't know that. That's one of the questions.
And if it does, what if there are implications about that? It just, and the other thing that occurred to me was this inevitable? Was this always going to happen eventually?
I think it probably was. I think it probably was. Jennifer Doudna had herself said that she had sort of waited for the day she would wake up and hear that someone had used this technique. So, certainly the people who are experts in the field were just waiting for that moment. There's a lot of hostility within the medical world, the research world, and so on to ethicists, because ethicists are perceived as kind of
getting in the way.
getting in the way. And look, in many ways, I feel their frustrations, and I see, that in many ways, ethicists and ethical guidelines absolutely can act as a barrier to the conduct of research. Now, I think some of those things are, when you look at the impact of things like the bureaucracy of ethics committees and so on, some of those things I think very much can get in the way of researchers being able to get on with the job. But I think what researchers need to understand is they've also got to take a step back and recognize that, for their work to be embraced, for society to support them in what they're doing, it is sometimes necessary to put the brakes on a little bit, to communicate what they're doing in a way that is accessible to the public, that is accessible to the people reviewing their research, and not to kind of, demonstrate hostility to that process, because ethicists are not wanting to stop good research. Ethicists are simply wanting to ask the questions which philosophers always ask, which is, what should we do? How, to think about important questions and engage with them and come out with the best answers to guide us to better society.
The other thing with it being HIV, though, I think, is his reasoning for choosing HIV is that in that particular area of China, there's a lot of stigma around being HIV positive or having AIDS, which is what the father of these daughters wanted to avoid, because that's the other thing. All of these couples were selected where the father had HIV and the mother didn't. I presume so that they're more exposed to HIV as a better test if they get infected, which is also kind of wrong. But by choosing HIV, his attempt was to not have them go through that social stigma of having HIV or AIDS. And that's a social problem. And it's trying to fix a social problem with a genetic technique, with a risky genetic technique, when there's no guarantee that they're going to get HIV anyway. If it was something that they were guaranteed to get, they were carriers of cystic fibrosis or something like that, where there's no other treatment for it, then it might be more acceptable to risk the complications that may come from this. But
yeah, that's a very, very good point. And in terms of this, using this kind of technique for, say, I don't know, let's say editing the genome of a family who have Huntington's disease, or the types of genetic diseases which are inheritable. That's a different question. But it's a different question, but it still has the same implications in terms of, first in human genetic experiments. And it's still at some level, if you look at what these particular couples have been asked to do, it's still preying on their fears and their hope to get them to consent to this.
Yep. The other issue I have seen is more from the researchers' side of things. There's a lot of researchers who are now worried that there's going to be a regulatory pushback as a result of this, that they're going to clamp down on what human genome testing can be done and what modifications we can see.
This taps into a little bit of what I was saying before about what he's done and what his team have done. It could have more of a chilling effect on the relationship between ethicists and researchers, where in fact, if they had engaged with this in an open way, in a transparent way, this essentially becomes an issue of trust. And we have enough issues with people trusting scientists, trusting their intent, trusting what they do, and he's really, really fundamentally interfered with people's trust in the intentions of researchers by doing this.
Yep. And for the record, I'm all in favour of using this technique once we are sure that it's safe and in the correct and ethical manner, because if we can eradicate HIV, if we can eradicate cystic fibrosis, Huntington's, all these other diseases, that can only be a good thing if we can do it safely and under the correct ethical manner. But this wasn't done like that, and that's deeply worrying and concerning.
Absolutely. One of the things that we're really working towards in research is about transparency. And you look at things like Ben Goldacre's work with the All Trials project, which is about researchers prospectively registering any clinical trial, which ensures that we can therefore track both the protocols used, how those protocols are changed, and then ensuring that researchers publish their results. Transparency in research is something that really is valuable and that we are kind of trying to promote, and he's really gone against that. So, from my point of view, I think this kind of research is incredibly important, but it's got to be done in the right way, and this just has not met that standard.
Yeah, so we'll keep an eye on it, and we'll see what happens when we actually see some more testable and interesting primary data from that. So he does say that he has submitted a paper to a journal for peer review and publication, but no one's seen it yet, so we don't know. Definitely one to follow. It'll be interesting. So, as always, all the links we talked about are in the show notes and on the web at scienceontop.com/319. And don't forget to check out scienceontop.com/donate to become a Patreon. And as of last week, we'll be donating all the contributions for 10 episodes to Penelope Green's favourite charities in her memory. Thank you for joining me today, Lucas, Penny and Joe.
Thanks, Ed.
Thanks, Ed.
Thanks, Ed.
And thank you everyone for listening. We'll be back again next week, putting science on top of the agenda. Join us then.
The world's people have spoken. Their message is clear. Time is running out. They want you, the decision makers, to act now. They're behind you, along with civil society represented here today, supporting you in making tough decisions, but also willing to make sacrifices in their daily lives. The people have spoken. Leaders of the world, you must lead. The continuation of our civilizations and the natural world upon which we depend is in your hands. Thank you.